Studies on the Fauna of Curaçao and Other Caribbean Islands, number 88, Social Behavior of the Paguridae and Diogenidae of Curaçao, by Brian A. Hazlett, Biological Laboratories, Harvard University. Introduction. The number of unanswered questions and untouched problems in the study of animal behavior is very large. This is particularly true in the subdiscipline of animal communications. The fact that the behavior of one animal can change the behavior of a second animal is too often only tacitly implied or stated without amplification. Before the language of any organism can be studied as a behavioral phenomenon, or the communication system of a phylogenetic series studied as an evolutionary phenomenon, it is essential that the basic behavioral biology of the species or group be examined and described. The present work is an attempt to fulfill this latter requirement. The social behavior of the marine hermit crabs, Paguridae and Diogenidae of Curaçao, North America, is described and in part analyzed. In addition to providing this basis, data have been gathered, which upon analysis have elucidated certain features of the aggressive communication systems of 8 species of hermit crabs. From the data gathered, the following aspects of hermit crab behavior are described. The behavior patterns of these crustaceans, as observed under laboratory and field conditions. The results of a number of experimental analyses of behavioral stimuli, and the results of some preliminary investigations of dominance hierarchies. The social behavior of hermit crabs has taken very little space in zoological literature. However, much has been written about the behavior and physiology of individual crabs from the viewpoint of shell habitation. See Rees, 1962a, 1963. Neurophysiology. Alexandrowitz, 1952. Steve, 1963. Sensory Physiology. Bonn, 1902. Goldsmith, 1918. Brock, 1927. Collar, 1927. Ten-Kate, 1930, and others. Symbiotic Relationships. Brock, 1927. Brightwell, 1951, 1953. Berner, 1953. And Feeding Behavior. Orton, 1927. Brock, 1930. Jackson, 1913, produced a rather good study of general anatomy and biology, as did Makarov, 1938, in his Introduction to a Systematic Treatment of the Russian Paguridae. Bouvier, 1892, and Bonn, 1903, briefly mentioned that hermit crabs do fight among themselves for shells. Ali and Douglas, 1945, observed that specimens of Pagurus longicarpus fight one another for shells and that there was a dominance order in this fighting, insofar as larger crabs always extracted smaller crabs. Their observations were confined mostly to interactions in which one individual was without a shell. They observed that the aggressive, shellless crab grasps the chellae and the legs of the occupant with one of its large pincers, jerks quickly, and so removes the smaller crab, and often throws it some distance away. Rees, 1962b, carried out more extensive observations of shell fights between individuals of Calcinus lavimanus, but again, one of the interacting crabs was artificially shellless. Reports of sexual behavior also have been few. Matthews, 1959, suggested that copulation in Paguridae is not necessarily confined to the short period following female ecdysis. He found spermatophores on immature females, the outer cuticular membrane of living embryonated ova attached to the pleopods, and on immature males. Matthews attributed this diversity of recipients of spermatophores to indiscrimination of the part of the male which proposes he may copulate with any hermit crab of his species that he can physically overpower. McGinty and McGinty, 1949, and Matthews, 1956, mention that male hermit crabs are sometimes seen dragging females about prior to copulation. Bott, 1940, reported that in Paguridae, a male remains with the female during the period of molting and until egg-laying. Brands, 1897, mentioned that the male P. Paguridae holds the tarsal segment of the second right ambulatory leg of the female with his left cheliped prior to copulation. Brands observed that during copulation, the major cheliped is held over the female in a protective fashion, as they both ease partially out of their shells. Coffin, 1960, noted that the male Pagurus samuensis held the shell of the female with his minor cheliped for a number of hours prior to the 1 second long copulation. The male waved the shell of the female back and forth, bumping it against his shell or body, and or twisting his body and shell while pressing against the shell of the female. During copulation, both eased out until only 2/3 of their abdomens remained in their shells. Camelveni, 1949, reported on the copulation of Clibanarius olivaceus. However, the observed pairs were without shells artificially, and their 90-minute pairing, with ambulatories intertwining, may have been aberrant. He also stated that recently molted females generally attracted the attention of males, but gave no details. Although the literature pertaining directly to the social behavior of hermit crabs is limited, many of the phenomena mentioned in this paper have been observed in other crustaceans. Douglas, 1946, reported straight-line dominance orders in the lobster, Homerus americanus. Bovbjerg, 1953, 1956, and Lowe, 1956, have investigated the development of aggression and straight-line dominance orders in several species of crayfish. Both size and sex, predominantly the former, were found to be factors in determining the order in a hierarchy. Although many authors have mentioned that members of a given crustacean species may fight with conspecific individuals, the manner of fighting is usually not described nor the signal display level of aggression recognized. One notable exception has been the work of Crane, 1941a, 1943, 1957, and 1958, and others on the conspicuous movements of the major cheliped of male fiddler crabs, Uca species. Crane has observed that these courtship movements signal territory holding to other males, and thus tend to keep them from intruding. In addition, Crane, 1958, has described a number of displays in Uca Maracoani, which she believes act as communicatory signals to conspecific individuals concerning the psychophysiological state of the displaying crab. Another group in which a signal display level of aggressive communication has been recognized is the snapping shrimp, Alphaeus, and Synalphaeus, Hazlett and Wynne, 1962. Both the sight of the open snapping claw and the jet of water produced by a snap act as retreat-causing stimuli, thereby avoiding actual physical contact between interacting individuals. Physical combats without damage are frequent among individuals of the common shore crab, Pachygrapsus crassipes, Hyatt, 1948, Bovbjerg, 1960, and the grapsoid crab, Dotilla nycteroides, Tweedy, 1950, Alcock, 1892, and Crane, 1941b, reported that burrow-occupying occipode crabs produce a twittering sound when an intruding conspecific individual enters its burrow. Presumably, substrate detection of this sound tends to induce retreat in the intruder. The presence of chemical attraction and/or excitation of males by ripe females has been supposed for a number of lower crustaceans. Pandylus Needler, 1931. P. borealis, Carlisle, 1959. Palaemonetes vulgaris, Birkenrode, 1947. Leander serratus, Forster, 1951. Crangon, Nouvelle, 1939. Orchestia gamarella, Williamson, 1954, but proven for none. A widespread characteristic among crustaceans is the restriction of time when mating takes place. In the great majority of species investigated, copulation is restricted to a short period following a female molt. Cancer pagurus and Carcinus manus, Williamson, 1902, and Neptunus pelagicus, Chapkin, 1956. Often the male crab carries the female about for hours or days prior to her molting, probably to assure that he is present when she is ready to mate. Carcinus manus, Brocuisen, 1937. Calinectes hastatus, Chidester, 1911, and Gamarus pungens, Holmes, 1903. The author wishes to express his gratitude to Dr. E. O. Wilson for his guidance, aid, and advice in connection with all phases of this work, and to Dr. W. H. Bossert of Harvard University for his assistance with mathematical problems and in programming the computer-analyzed portions of this study. In addition, thanks are given to Dr. A. J. Provenzano, Jr. of the University of Miami for confirmation of species identification, to Dr. Ingvar Christensen and the staff of the Caribbean Marine Biologics Institute, Caribbean Marine Biological Institute in Curaçao, North America, for their aid and hospitality, and to Miss Joanne Helsberg for aid in the preparation of the text. I also wish to thank the National Science Foundation, whose training grant in evolutionary biology, administered by the Department of Biology of Harvard University, was the source of travel and facility funds that made this work possible. Part of this research was carried out with the support of a National Science Foundation Fellowship, number 11372, and a National Institutes of Health Fellowship, 741 GM 14, 274 01A1. Portions of this work were presented to the Department of Biology, Harvard University, in partial fulfillment of the requirements for the degree of Doctor of Philosophy. Figure 1. Location of collecting study areas around Curaçao, North America. Collecting areas on Curaçao. Curaçao, NA, is a medium-small island with an area of about 430 square kilometers, about 65 kilometers off the coast of Venezuela, Figure 1. A constant wind from the east maintains strong wave action on the northeast side of the island. Even those sheltered bays and inlets present here are, for the most part, characterized by heavy wave action. In addition, the inshore bottom is made up of relatively smooth, large, flat rocks, e.g. Boca Grande and Bartul Bay. The majority of the collecting study areas were on the south coast of Curaçao. The greatest amount of work was done in the shallow waters directly in front of the Caribbean Marine Biological Institute, Carmabi, adjacent to Piscadera Bay, which may be called Piscadera Buiten Bay, Piscadera Outer Bay. Wave action there is moderate, and the gently sloped bottom is composed of detritus-covered rocks and coral rubble. This rocky condition extends out for 15 to 20 meters from shore. There is then an area of sandy bottom which gives way to coral formations at the edge of a sharp drop-off. The drop-off starts at a depth of about 8 meters and descends rapidly. The entrance to the Piscadera Binen Bay, Piscadera Inner Bay, is a relatively shallow channel, less than 3 meters deep at one point. It has steeply sloping sides made up of small to medium, rather clean rocks, which offer refuge to many organisms. As the tide goes out and until it rises again, the channel and the area in front of and to the west of the entrance of the bay are greatly discolored by the emptying of the inner bay. This tidal movement surrounds the organisms in the entrance in a relatively thick suspension of organic matter. Piscadera Binen Bay is a large body of water, covering an area of about 0.6 square kilometer, with an average depth of about 3 meters. Due to the concentration of civilization around it and the topography of its narrow entrance, the bay has a rich suspension of organic particles in its waters. The majority of the shores of the inner bay is composed of mangrove mudflats which are partially exposed at low tide. The entrance to Spans water is much wider and deeper than that of Piscadera Bay. It is about 35 meters wide and over 10 meters deep. The eastern shore is very steep and made up of medium to small rocks and pebbles. The western shore is relatively gently inclined from about 8 meters upward. Proceeding inward, the bottom of the west slope changes from a very large dense growth of elkhorn coral, Acropora palmata, to a shallow flat of eelgrass, to a clear sandy bottom. Spans water itself, that is, its inner bay, is a 3.5 by 1.5 kilometer large body of relatively clear water. Its inlets, such as Bracaput, are shallow mangrove mudflats, but the water is clearer than of Piscadera Bay mudflats. The entrance to Saint Marta Bay is also steeply banked and made up of small pebbles and rocks. The area just outside the bay is a shallow shelf with heavy coral growth and relatively rough surf action. Versen Bay is a moderately sloped open inlet. The bottom is covered with medium-sized detritus-covered rocks. Surf action is moderate. Boca Saint-Michel, St. Michael's Bay, is an open area with medium-light surf action. The first 5 to 6 meters of the bottom extending out from the waterline is a broad, gently sloped rock shelf covered with detritus and algal growth. A number of artificial piles of large rocks project seaward on this shelf. At about 1.5 meters water depth, the shelf drops about 50 centimeters to a sandy bottom, which extends out to rather deep water. Relationships of the hermit crabs studied. Relationships of the hermit crabs studied. The species studied in this investigation were identified by Dr. A.J. Provenzano, Jr., of the Marine Science Laboratory of the University of Miami. Specimens of the species studied have been deposited in the Museum of Comparative Zoology, Harvard University. MCZ numbers 12848 through 12858. These decapod crustaceans are members of the section Anomura. The systematic classification used in this paper is that proposed by MacDonald, Pike, and Williamson, 1957, based upon larval studies. This classification has been supported by further larval studies by a number of workers. Provenzano, Personal Communication. The marine hermit crabs, characterized by poor calcification of the cephalothorax and the uncalcified asymmetric abdomen, are divided into 2 families. The Diogenidae may be distinguished by the fact that the bases of the third pair of maxillipeds are close together, and the chelipeds are either equal or the left is larger than the right. In the Paguridae, the bases of the third maxillipeds are far apart, and the right cheliped is usually longer than the left. The Diogenidae are more characteristic of shallow tropical waters, while the Paguridae are more characteristic of deeper and colder waters. Important differences in larval morphology also separate the families. Among the diogenid genera encountered in this study, the genera Clibanarius and Calcinus are more closely related to one another and distinct from Paguristes. Within the genus, Clibanarius tricolor and Antillensis are more closely related to one another, as are Clibanarius cubensis and Vitius. The Paguridae includes, among many other genera, Pagurus and Pilopagurus. Pagurus miamensis and Pygmaeus are more closely related on morphological grounds than the other 2 species of this genus encountered in this study. Makarov, 1938, mentions the possibility that a form similar to Pilopagurus may have given rise to the stone crabs, the Lithodidae. According to MacDonald, Pike, and Williamson, 1957, and others, the Lithodidae are derived from some pagurid form. A taxonomic note which needs to be presented in the literature once again is the correct use of the generic names Pagurus and Eupagurus, as pointed out by Provenzano, 1959, and others. Opinions by the International Commission on Zoological Nomenclature have asserted the incorrectness of the use of Eupagurus. This name is a junior homonym of Pagurus fabricius, 1775. Despite the mention of this situation in several articles, some workers continue to use the incorrect name Eupagoras.